Okay, good evening, you all, to the live session of experimental biochemistry. I am Surasree. I am a Prime Minister's Research Fellow working in the Department of Biotechnology of IIT Kharagpur. And through these 12 weeks, I will be one of your teaching assistant, and we will be solving and discussing many problems related to the course of experimental biochemistry. So, please let me know if I'm audible to all of you. Then we can begin today's session. Please, someone let me know if I'm audible. Okay. Alright, so in today's session, we will cover some of the few topics which were discussed in the week 1 of this NPTEL course, which will include accuracy and precision, acids, bases and pH, and the last one is buffers. So if you have gone through the lectures and the video demonstrations of the week 1 of this course, you had gone through these topics. The first one is accuracy and precision. So what are we discussing when we are discussing about accuracy and precision is that we are discussing about values, right? So measurements of values. So in this particular course, which is experimental biochemistry, which means the experiments that we do in biochemistry laboratory, they involve various measurements of, for example, volume, measurements of pH, measurements of weight, measurements of temperature, so on. And while we are measuring these values, any parameters, while we are measuring, it is important to keep in mind that these measurements have to be accurate and precise. So these terms may mean very similar in colloquial language, but accuracy and precision, scientifically, they are quite different terms. So accuracy is the closeness of the estimated value to that which is considered to be a reference value. So what is this reference value? So for example, we have taken a volume of liquid and we know that this volume is 10 ml. Okay, and if then we measure the volume of the liquid and we find it to be 9.98 ml or approximately 10.01 ml, and from that we can say that therefore our measurement is accurate because it is close to our reference value or to the value which we assume to be true, right? So that is called accuracy. And what is precision? So now suppose now you are measuring the volume of a liquid for 3 times. So you are measuring the volume of the same liquid 3 times, and if you are measuring 3 times, you will get 3 different values while doing the measurement, because while we are doing any measurement, it is there will be some human error, there will be some instrumental error. So 3 different values we will observe in 3 different experiments. So suppose we are taking the measurements and it is like 9.99 ml, 10.01 ml and 9.98 ml. So this is this is one set of the values that we observe. And suppose your friend is also measuring the same liquid, same volume of liquid, and suppose that friend gets these values: 9.88 ml, 10 ml, 9. 9.87 mm, right? So this is measurement done by A, this is measurement done by B. Now precision is the reliability of the result. That means when we are repeatedly testing one particular one particular experiment, or when we are repeatedly taking measurements of one particular experiment, then we have to check how close our values are, so that we can say that yes, my experiment is reliable, or my data that I'm getting is reliable. So for the reliability of the result, the values should be close to each other, right? So that is called precision. So now can anyone of you tell me, like, which one of the values you think to be precise, like for the values of A or the values of B, more precise? So precision is when the values are close to each other. So in which set do you think the values are more close to each other? Can anyone tell me? So the more precise set of results here is this set A, because you can see that the three values are 9.99 ml, 10.01 ml and 9.98 ml. So these three values are quite close to each other than the set of B, right? So that is why the set of A is more precise. Now if you look at this particular diagram, you will see that when the values are, so suppose what is your reference value, or what is the value that should be that you should observe, that is the standard or the reference. Suppose it is in here, this is the standard, so that is the bullseye, right? So now here in this particular diagram, you can see that all the values are scattered, and so if they are not close to each other, they are not close to the standard value as well, so they are less accurate and they are less precise. Whereas here in this diagram, you can see that these values are very close to each other, but they are not close to the standard or the reference value. So therefore they are more precise, but their accuracy is low. Whereas here in this third diagram, you can see that these values are not close to each other, but they are close to the standard value, which is why it is a high accuracy but low precision condition. So the values are not close to each other, but they are close to the standard. And this is the ideal situation where the values are close to each other and also the values are close to the standard or the reference. So therefore it is a high accuracy, high precision condition. So in our experiments, we should try to attain our results which are reliable. That means after repeated experiment, we are getting the same value or the same result, which means our result is precise, and we also should be able to achieve accuracy. That means we should be able to reach close to the right value or the value which is a standard value, right? So that is the difference between accuracy and precision. Now in the next topic, we will come to acids and bases and also pH. So in class 11th and 12th chemistry, You must have learnt about acids and bases. And what is an acid? An acid is a proton donor. So, for example, HCl, what it does when it dissociates into hydrogen ion and chloride ion. So, an acid is known to be a proton donor, whereas a base is a proton acceptor, or it is also called an hydroxide ion donor. So, for example, potassium hydroxide KOH, when it dissociates, it gives K plus and OH minus ions, right? So that is acids and bases. Now we will come to pH and its measurement. Now what is pH? pH is a measurement of the acidity or basicity of any solution. So pH is basically the measurement of the concentration of hydrogen ions present in any dilute solution. So it measures the concentration of hydrogen ions. And what is the formal definition of pH? The formal definition of pH states that it is the negative logarithm of hydrogen ion activity. Okay, it can be written as this. pH is equal to minus log of H plus ions, or the concentration of H plus ions. Now pH can be measured on a scale of 0 to 14. So please someone tell me, like on the scale of 0 to 14. So if this is a scale of 0 to 14 and 7 is the middle value, then please someone tell me what is the pH range of the acids and what is the pH range of the bases. Someone please switch on your microphone and tell me. What is the range of pH of the acids and what is the range of pH of the bases? Come on, all of you know that. Please tell me. Sharmila, can you tell me? This is an interactive session, so the more you interact, it will be better for all of you. So you are free to interact. This is not one-way traffic, please remember, because the lectures that you are seeing on NPTEL website, those are recorded lectures. That is actually a one-way traffic. But here in these sessions are meant for interaction. So if you are free to interact, you are free to ask me any question, and you are free to answer anything. Okay, so the range of pH of the acids is from, yes, and what is the range of pH of the bases? Yes, thank you. and 7 is, what is it called? 7 is the, yes, 7 is the neutral pH. Right, so now next question comes, how do we measure pH? If pH is the measurement of the negative logarithm of hydrogen ion concentration, then how do we measure pH? So there are several methods by which we can measure pH. One is a pH meter, another is a pH paper. Now these two are very different in their application, because a pH meter is an instrument where there is an electrode which measures the pH. So there's a glass electrode which can measure the pH, and how it measures the pH I'm telling you in the next slide. But the pH paper is a paper where which actually gives you a particular color in a particular pH solution, right? So if you take a drop of a particular pH solution or a particular solution and pour it on the pH, on a strip, pH strip, on a small section of the pH strip, it will show you a particular color, and based on that color you will be able to approximately say what is the pH of that particular solution. So here you can see that the different pH are shown in different colors, right? So that is the standard. And suppose you are taking a solution of pH 6 and you do not know that it is of pH 6, but you pour it, you just put a drop of that solution on the pH paper and it shows this kind of green color, then you will be able to say that yes, this solution is of pH 6. But obviously pH is it is not only an integer number, right? So it is not a whole number only. So pH can be 7.2, pH can be 9.3. So this kind of variation cannot be observed in or cannot be measured by pH paper. So which is why you need a pH meter for more accurate and more precise measurement of pH. Okay, so for example, if your solution is of 7.4 pH, in the pH paper you will see a color which is intermediate of pH 7 and pH 8, but you won't be able to say whether the solution is of pH 7.4 or 7.5 or 7.7, but that you can test using a pH meter. Now coming to a pH meter has these components. Okay, pH meter has a pH measuring electrode, a reference electrode, and a high input meter. So what does this electrode do, the measurement electrode? So there is a voltmeter in this pH electrode which measures the difference between the voltages of the measuring electrode and the reference electrode, and the difference in these voltages is measured by this voltmeter, and then there is a meter. So this is the display, and this is where the voltage difference is translated into pH. Okay, so there's a scale which translates the voltage difference into pH, and then it displays it on the screen. So this one is the electrode, and this is a glass electrode. And remember that even though pH meter is very accurate and it's very precise, but it is also difficult to maintain a pH meter. Why is it difficult? Because you need to take care of the glass electrode. You need to make sure that the glass electrode is stored properly, and when you are using the glass electrode, you should rinse it properly with water, with double distilled water. And also when you are measuring the pH, you should not there should not be any crack on the glass electrode, otherwise the electrode will be damaged. So we need to take care of the electrode, and we need to make sure that the electrode does not get coated in any kind of solution, because we will be measuring pH of different kinds of solutions. So we need to make sure that the glass electrode is not coated. Okay, so there are certain errors which might arise because of these defects. That is, there can be asymmetric potential in the glass electrode, there can be aging and coating of the electrode, and there can be clogging of the electrode diaphragm. So what do we need to do to make sure these does not happen? We should rinse the electrode properly, we should store it in the proper solution. So usually glass electrodes, pH electrodes, are stored in 3 molar or 4 molar potassium chloride solution. So we should properly dip the bulb of the glass electrode in the solution while we are storing it, and we should always rinse it properly, wipe it with like dry tissue paper, and we should make sure that there should not be any layer of solution around that glass electrode. Now there can be one other problem also in a pH measurement experiment, that is, if your pH meter is not calibrated properly. So what is that? What is this calibration? So it's like you have to make a standard, or you have to adjust the pH meter so that it can recognize the standard solutions of known pH. So what is actually done here is that there are solutions which are provided by the manufacturer, which are solutions of pH 4, pH 7 and pH 10. So these are solutions which we can manufacture, and these are of known pH. And now what we do is, when we calibrate the pH meter, we dip the glass electrode in suppose solution of pH 4. So the standard solutions, they are of pH 4, pH 7 and pH 10. So this is a 3-point calibration. Okay, so we dip the solution in pH 4 and we allow the pH meter to calibrate. So what happens here is that it measures the pH, okay, right? And suppose when we have dipped it in the solution of pH 4, it shows the pH of pH 4.3, right? But we know that this is a solution of pH 4, so the pH meter must display pH 4, not pH 4.3, right? So therefore we have to calibrate the pH meter accordingly. So what we do is, then we allow the pH meter to calibrate and we enter this value of 4, right? And then the pH meter will adjust itself according to that, and it will now show the value of pH 4 in a pH 4 solution. Now next what we do similarly in the pH 7 solution. So we dip it in the pH 7 solution and we allow it to calibrate. So now the pH meter shows pH 7 in this particular solution. And similarly we do it for pH 10. So in the acidic pH, in the neutral pH and in the basic pH, 3 points we take and then we calibrate it accordingly, right? And once this calibration is over, then we can easily measure the pH of our required solution. Okay, so it is always recommended to calibrate your pH meter before you are measuring the pH of any unknown solution. Okay, so these are some of the cautions which you should take while using a pH meter. And these kind of problems do not arise in a pH paper because it is very easy to use, it is very cheap, it does not require much effort for maintenance. But pH meter is very accurate, but it requires these kind of maintenance. The principal maintenance is that of the glass electrode, that should be properly stored and cleaned. Now next, coming to the next topic of us, which is buffers. So you must have heard about buffers. Buffers are the solutions which can resist changes in pH when small amounts of acids or bases are added to them, right? So suppose you have taken a particular solution which you want to keep in a particular pH. Now, why is this pH required? Why do we require the pH to be specific, or why do we require the pH to be constant? Now, in biochemistry experiments, what do is that we try to mimic the cellular environment or the environment which exists in our physiological condition, right? So anyone of you tell me what is the pH of blood, or what is the pH of our body fluids? Very good. So our blood pH is approximately 7.4, or the pH of our the body fluid or tissue fluid is 7.4. So all the reactions which are carried out in this blood or in our tissue fluids, they are carried out in this particular pH. But that is also not not always true, because you know that in the digestive system there are many enzymes which work in low pH or alkaline, in acidic pH or alkaline pH. So for example, have you heard of these protease enzymes or protein digestic enzymes, like for example pepsin? Pepsin works in a very low pH condition, okay, so in an acidic pH, whereas like chymotrypsin or trypsin, these are protein digesting enzymes which work in high pH conditions or alkaline pH conditions. So there is an optimum pH for each enzyme. Optimum pH. So this is the pH where the enzyme will be most functional. So this optimum pH exists for all enzymes, and not only that, there are several biomolecules which are also dependent on the pH. So they need to be present in a stable pH. For example, our DNA, nucleic acids, our other molecules, like for example proteins, they should be present in a particular pH, otherwise they will not be functional. So to maintain their structure and function, there should be a constant pH maintained in our body. And if we are doing any biochemical experiment, and if we are working with, for example, proteins or nucleic acids, we should also maintain that pH in our in vitro conditions, right? In vitro means when we are doing the experiment in tubes, right, outside the body or outside any organism. So therefore we should maintain that pH condition in our in vitro systems as well, which is why we need buffers, because buffers can maintain the pH at the specific value, because even if small amounts of acids or bases are added to it, it does not change, it resists the change in pH. How does it resist the change in pH? We will discuss it later on. But remember that buffer is a mixture of weak acid and its conjugate base, right? So what is a conjugate base? For example, if HA is an acid, when it dissociates, it gives H plus ion and A minus conjugate base. So this is the conjugate base. So if HCl is the acid, then Cl minus is the conjugate base, right? So buffer is a mixture of weak acid and its conjugate base. Now, some of you might have heard about this equation, Henderson Hasselbalch equation, which is one very, very important equation in the measurement of pH and in all the related measurements of buffers and how to maintain a pH. This equation is very, very important. So how do we reach to, or how do we derive Henderson Hasselbalch equation? So this is basically related to the pH of the solution, how the pH can be determined when a weak acid is dissociating. So what happens here is that HA, suppose, is a weak acid. So I told you when it dissociates, it forms H plus ion and A minus conjugate base, right? So now what is Ka? Ka is the dissociation constant of this weak acid. Now, and Ka, we can write it like this. Ka can be written as a product of H plus and A minus divided by the concentration of the HA, right? So that is the dissociation constant of this acid. And now if we put negative logarithm of this whole equation, what we get is that minus log of Ka equals to minus log of H plus minus log of A minus plus log of HA, right? And we know that minus log of Ka is pK, minus log of H plus is pH, right? So putting these two values here, we get pK equals to pH minus log of A minus divided by HA. Okay, and if we change the direction, change the sides here, we get this particular equation, which is pH equals to pKa plus log of concentration of A minus divided by concentration of HA. So it is nothing but pH equals to pKa plus log of concentration of conjugate base by concentration of acid. This is the particular equation, which is the Henderson Hasselbalch equation, and we will further use it for determination of pH of any buffer. Okay, so this equation is very important, and you have to remember this equation across all biochemistry lessons. In all chapters, this might be a very important equation. Now coming to one crucial event or a crucial component of the buffers, which is the buffering zone. Now what is a buffering zone? So buffering zone is referred to as this, which is a pK plus minus 1 pH unit. So suppose an example is acetate buffer. Acetate buffer has a pK which is 4.8. Okay, acetate buffer has a pK which is 4.8. So that means what will be the buffering zone? What is the buffering zone? First of all, the buffering zone is the zone where, or the range of pH where the buffer is most effective. That means where it can resist the any small addition of acid or bases. So it can resist the change of pH. So the most effective range of buffering action is the buffering zone. Okay, and I told you that the buffering zone is pK plus minus 1 pH unit. Now from where do we infer that? So before discussing that, let's first discuss, so therefore what will be the buffering zone of the acetate buffer? Can anyone tell me what will be the buffering zone of the, or the buffering range of the acetate buffer? Buffering zone is pK plus minus 1 pH unit. So greater than 1, from pK and less than 1 from pK. That is the zone, or that is the range of the buffer, which means that the buffer will be most active or most efficient in this particular zone, right? So Orkopal is saying that it is 3.8 to 5.8 exactly. So we can say approximately from 4 to 6, right? So that will be the buffering zone of the acetate buffer. So which is from because the pK is 4.8, right? So now let's come into this question, that why are we considering plus minus 1 pH unit as the buffering zone? Why is it important? Because if we now look at this equation once again, that is the Henderson Hasselbalch equation, which is pH equals to pKa plus log of concentration of conjugate base divided by concentration of acid. We find out that, so when pH equals to pKa, if we put it here in this particular equation, we will find out that when pH equals to pKa, log of this will be 0 when pH equals to pKa. So pH minus pKa becomes 0. So log of A minus concentration divided by HA concentration becomes 0, which means that A minus by HA concentration becomes 1, which also means that concentration of the conjugate base becomes equal to the concentration of the acid, right? So therefore when pH equals to pKa, we can say that the acid concentration will be equal to the concentration of the conjugate base. That is when pH equals to pKa. Now what happens when pH is less than pK? So for example, if we consider this, that when pH is less than pK, then what will happen is that this will be a negative value, and therefore we will get HA, concentration of HA will be greater than concentration of A minus. So the acid part will be more dominant. The uncharged part, or this acid part, will be more dominant than the conjugate base. Okay, so the concentration of the acid will be much more than that of the conjugate base. And suppose, so that means this acid is not completely dissociating, right? So suppose there is a very little dissociation of this acid, which is why the conjugate base concentration is not similar to that of the acid concentration, right? So that means it is not in the equilibrium position. So there what happens, suppose there is a I'm erasing this part. Suppose we say that there is 10% dissociation of the acid. So if dissociation is 10%, that means the concentration of A minus divided by concentration of HA will be 10 by 90, because 90% of the acid is not dissociated, right? The only 10% of the acid is dissociated, so it will be 1 by 9, right? And if it is 1 by 9, then what happens is that it is log of 1 by 9 is approximately, can anyone calculate what is log of 1 by 9? It is approximately minus 0.95, or we can say approximately minus 1 if there is 10% dissociation of the acid. This is one condition, and the other condition is when there is approximately 90% dissociation of the acid. So what happens here is that if there is 90% dissociation, then this ratio will be reversed. So this will be A minus by HA will be 90 by 10, which is 9, right? And log of 9 will be approximately plus 1. So we can see that when there is 10% dissociation and when there is 90% dissociation of the acid, the range of the pH value will be when there is 10% dissociation, the pH will be pK minus 1. And when there is 90% dissociation, the pH value will be pK plus 1. So you can see here why we are coming to this particular value, that is buffering zone is when pH will be plus minus 1 of pK, because when there is 10% dissociation of the acid, we will get the pH as pK minus 1, and when there is 90% dissociation, that means almost complete dissociation we can say, then the pH value will be pK plus 1. So therefore the buffers will be most effective in this range, which is pK plus minus 1. So from minus 1 to plus 1. Is that clear? Any doubt? And of course, when pH value will be greater than pK, then this acid concentration will be lower than the conjugate base concentration. So any doubt regarding this particular part where we are saying that the buffering zone is when the pH will be plus minus 1 from the pK value, and which is actually the buffering zone. And how do we determine that? We determine that using this Henderson Hasselbalch equation, where if there is 10% dissociation of the acid, the pH will be approximately pK minus 1. If there is 90% dissociation of the acid, then the pH value will be pK plus 1. Alright, if there is no doubt, you can write in the chat box if you have any doubt, or if there is no doubt, we will proceed. Okay, one more time should I repeat? Alright. So I'm creating a new notebook and I'm showing it to you. So pH is pK plus log of base by acid, or you can say that conjugate base by acid. So this is the Henderson Hasselbalch equation, right? Please tell me whenever there is any doubt, you can interrupt me in between, okay? There's no problem, you can always interrupt me and you can always ask. You can unmute your microphone and ask. Alright, so this is the Henderson Hasselbalch equation. Now we are saying that the buffering zone is plus pK plus minus 1. But before going to the buffering zone, let's consider there is a weak acid, right, which is for example HA. Alright, so now when this weak acid is dissociating 10%, so suppose there is 10% dissociation, and we have to predict, or we have to calculate what will be the pH if the acid is dissociating 10%. Alright, so how do we calculate that? pH equals to pK plus log of base by acid. Now HA, when it dissociates, it gives H plus ion plus A minus ion, right? A minus is your base and acid is this one, right? So you will get pKa plus log of A minus divided by HA, right? If there is 10% dissociation, that means only 10% of A minus will form and the rest of the acid will be in the form of HA, which will be 90%, right? So if we put the value here, we'll get A minus as, the value of A minus will be 10 and the value of HA will be 90, right? So now if we calculate it, it will be log of 1 by 9, which is approximately minus 0.95, or we can say approximately pKa minus 1. So when there is 10% dissociation of the acid, the pH value is approximately pKa minus 1. Is that clear, this part? If it is clear, then only I'll proceed. Okay, all right. So now we are considering the next situation, which is 90% dissociation. So if there is 90% dissociation, of course the A minus will be 90 and HA will only be 10%, right? So pH will be equal to pKa plus log of, I'm writing it again, A minus by HA concentration, which will be pKa plus log of, if there is 90% dissociation, 90 by 10, which is pKa plus log of 9, which is approximately pKa plus 1. Okay, so this is the pH you will be getting of the solution when there is almost 90% dissociation of the weak acid or the acid, right? So the pH value, therefore you can see it varies between pKa plus minus 1, right? Plus 1 from pKa and minus 1 from pKa. So this will be the zone where the buffer will be the most effective, which is why it is called the buffering zone. Go through it, tell me if this is okay. Alright. Okay, then we will proceed. So now let's come to buffer action. So I am talking about the buffers resist the change in pH, right? So how do the buffers resist the change in pH? So now if we consider this particular buffer, which is an acetate buffer, so it is acetate buffer when so it is composed of this weak acid, acetic acid, and its conjugate base, which is sodium acetate, right? So now weak acid, acetic acid will dissociate into acetate ion and H plus ion, and the weak base, not the weak base, the conjugate base will dissociate into acetate ion and Na plus ions, right? So now how do these buffers prevent the change in pH? So suppose if you add few drops of acid in it, so that means the acid will add proton ions into it, right? The H plus ions will be added. Now if H plus ions are added in this system, then what happens? The equilibrium is shifted to the left, right? The equilibrium is shifted to the left so that these additional H plus ions can be absorbed, right? So that is why there is a shift of equilibrium towards the left side, and there is absorption of the H plus ions so that the H plus ion concentration in the solution remains fixed, and therefore there is no change in pH, right? that is how the buffers are resisting the change in pH when we are adding acid in it by absorbing proton ions, so that there is the net proton ions in the solution remains fixed, and so the pH remains fixed, because the pH is dependent on the concentration of the proton ions or the H plus ions, right? Now what happens if we add base to it? If we add, suppose, hydroxyl ions or these OH minus ions in this particular solution, then what will happen? Then this OH minus ion will react with this H plus ion and it will form H2O, right? So therefore these OH minus ions will be neutralized by these H plus ions by forming water, and therefore the H plus ion concentration, then the H plus ion concentration will diminish. If that occurs, then the pH will also change. So the buffers cannot let that happen, right? So therefore in this system the equilibrium will be shifted towards the right by releasing more and more protons, right? So the equilibrium in this case will be shifted to the right, so that this acetic acid will release more and more proton ions, so that again the hydrogen ion concentration or the proton concentration in the solution can remain fixed and there is no change in pH, right? So that is how the buffers are acting, either by releasing more and more protons when bases are added, or by absorbing protons when acids are added, right? So that is how buffers are acting and resisting the change in pH. Is that fine? Any doubt? Okay, if there is no doubt, then we will proceed to the next part, which is the buffer capacity. So it is the thing that I'm trying to say multiple times, that is resist the change in pH. So it is the ability of the buffer to retain its pH when strong acid or base is added, and it corresponds to the amount of hydrogen ions or H plus ions or OH minus ions which can be neutralized by the buffer. So that is the capacity of the buffer. Whether it's a strong buffer or whether it's not a good buffer, that is determined by this buffer capacity. Now there can be several ways by which we can prepare a buffer. So first thing is titration method. So suppose we are taking two components, one is acid, one is base, and what we do is that one component is added slowly in the other solution until the required pH is obtained. So this is a crude method, and because the amount that we are adding, that we have to add, is not fixed. Okay, so that is why it is called a crude method, and we cannot calculate, or suppose we are not calculating how much of the acid or base is required. So this is how, but this is a very common way of preparation of buffers, and this is how we prepare phosphate buffer in our laboratory. And if you have gone through the phosphate buffer preparation demonstration, you might have seen that there are two components in the phosphate buffer. One is an acidic component, which is NaH2PO4, and another is a basic component, which is Na2HPO4, right? So what we do is, suppose we have to prepare 100 millimolar of phosphate buffer. 100 millimolar of phosphate buffer we have to prepare, and what we do is that we prepare 100 millimolar of NaH2PO4 and also 100 millimolar of Na2HPO4, right? And then what we do is that these two have two different pH. So NaH2PO4 has a pH of approximately 4 and Na2HPO4 has a pH of approximately 9. So you can see that which one is acidic, which one is basic. Now what we do is, suppose we have to prepare the 100 millimolar of phosphate buffer of pH 7. So in these two, which one is closer to 7? The pH 9 thing, right? The Na2HPO4 is closer to 7 pH. So we take that. We take this Na2HPO4 and we add drops of this NaH2PO4. And by adding drop of these NaH2PO4, we measure the pH continuously, and we keep on adding this NaH2PO4, and once the required pH is obtained, we can collect the buffer and we can use it in our experiments, right? So this is a titration method of preparation of buffer. Now next, another method of preparation of buffer can be by calculating from the pKa by using the Henderson Hasselbalch equation. So what we do here is that suppose we have to prepare 500 ml of 0.1 molar of acetate buffer using 0.1 molar of acetic acid and 0.1 molar of sodium acetate, and the final pH should be 4. So this is the final pH. Now what we do here is that we have the two components, which is 0.1 molar of acetic acid and 0.1 molar of sodium acetate. So their molar strength is same, right? And we have to also prepare the buffer, which is of 0.1 molar. So the molar strength are same for all the components. Now we have to prepare the buffer of pH 4, and our pKa of acetic acid is provided as pH 4.8. pKa is provided as 4.8. So now how do we prepare the pH 4 buffer? So now we will put these values in this particular equation. So pH is 4, our required pH, pK is 4.8 plus log of A minus by HS. So this is our concern, right? How much of the base and how much of the acid we have to add to prepare this buffer of pH 4. So that is what we are going to calculate from this equation. So this will be log of A minus by log of HA will be minus 0.8, right? So what we can write here is that A minus by HA is 10 to the power minus 0.8, and if we calculate it 10 to the power of minus 0.8, we get approximately 0.158, I guess. Yes, we get approximately 0.158. So now we know the exact ratio of the acid by or base by acid, right? 0.158, by which we should add the base and acid to get the pH 4 buffer, right? So this approximately gives you the, we can also approximately write that HA by A, or the acid by base, will be 6, right? So therefore we have to add 6 times of the acid than that of the base, right? So suppose we have to prepare 500 ml of the acetate buffer. What we can do is we can take 70 ml of the acid, which is the 0.1 molar acetic acid, and we have to take 6 times 0.1 molar 70 ml of 0.1 molar of the sodium acetate, and we have to take 6 times the acid, right? So 420 ml approximately of 0.1 molar of acetic acid. So is that clear? And then we add water to adjust the volume, and then we add some final pH adjustment might have to be done, which will be very less also. Very less pH adjustment might have to be done. So here what we are doing is that we are calculating how much acid and how much base we have to add, so that we have a clear idea of what is the ionic strength of our final buffer. But what we are doing in this titration method was that we did not know what is the ionic strength of the final buffer, because we are adding unknown amounts of acid or base into it while we are adjusting the pH. There can be another method of buffer preparation, which will be like referring from the tables which are already prepared by biochemists, and they are available in biochemistry books. So referring from the table, we can get the exact amount of base and acid we have to add, and then how much pH we have to adjust, or if we have to adjust some pH. Okay, so these are several methods of buffer preparation. Now coming to the commonly used buffers. One is phosphate buffer. A very commonly used buffer in biochemistry experiment is phosphate buffer in the range of pH 6.5 to 7.5. But the problem is that in phosphate buffer, phosphate is usually present in cells, right? And so it provides a very physiologically similar environment, but the problem is that this may inhibit some enzymatic reactions as well. So that is the problem of phosphate buffer, but it has a very high buffering capacity, and it is water soluble also. Other buffers are like, for example, carboxylic buffers, borate buffers. Carboxylic buffers, for example, acetate buffers, citrate buffers, they can be acting in the pH range of 3 to 6. And borate buffers act in the pH range of 8.5 to 10. But remember, these carboxylic acid buffers, borate buffers, they provide very the problem with these type of buffers is that they can interact or form complex with several biomolecules, and then therefore interrupt or interfere with the reaction that we are trying to measure or observe, right? So that is the problem of these kind of buffers. Now another type of buffer which are known as goods buffers. So these are Zwiiterionic buffers. So you know what is a Zwiiterion, right? So this kind of buffers developed by Norman E. Good. So what is the principal advantage of this goods buffer is that these buffers are synthetic, so they usually do not interact with any of the biomolecules. So they do not form complex with the biomolecules, they do not inhibit any enzyme reaction. But the advantage is also that they have very high buffering capacity, and they can be suitably used for various biochemical reactions. So for example, MES, HIPAS, TRIS, TAPS, malate, these are some of the Zwiiterionic buffers which are commonly used. For example, coming to TRIS buffer, this is a very commonly used buffer in biochemical reactions, and this is tris hydroxymethyl aminomethane. That is the full form of TRIS. This is how the TRIS looks like, and the thing is that here you can see there is a amine and there are 3 hydroxyl groups, right? So there is a lone pair on this nitrogen, right? But this lone pair is less available because this lone pair is pulled by the 3 hydroxyl groups, right? So which is why it lowers the pK of the tris, which is approximately 8.1. So pK of tris is approximately 8.1. So you can assume what will be the buffering range, approximately from 7 to 9, right? So it is a suitable buffer near neutral pH. So pH range will be 7 to 9 because the pK is 8.1, right? You might have gone through the video demonstration of how tris buffer is prepared. So we take molar, like if we are preparing, for example, 1 molar of tris buffer, then we weigh the required quantity of the tris powder, we dissolve it in water, and then we adjust the pH by concentrated hydrochloric acid, right? Next is HIPAS buffer. This is also again very commonly used used for biochemical reactions. It is 4,2-hydroxyethyl 1-piperazine ethane sulfonic acid, and its pK value is between 6 to 8. It is also very highly soluble and does not react with biochemical reactions. So this is also chemically and enzymatically stable and easy to prepare. So while I'm discussing about these buffers, you might have understood that what are the several features or characteristics of these buffers. These buffers should be such that they have less interaction with the biomolecules. They should not inhibit any enzyme actions. Then, for example, they should be stable. They should be water soluble, and they should be active over like there should be a buffering range, which is like determined, which is known, right? So these are the several characteristics of a buffer which we can use. Now, all right. So, anyone having any doubt over this point? All right. If you do not have any doubt, then we may proceed to our question answers. So problem discussion part of this of these live sessions, and so I will share with you many problems, and please let me know the answers in the chat box. Okay, let's start. So the first question is, pure water is neutral, and what is the reason? Please let me know your answers in the chat box. Okay, so I can see that most of you are saying the answer is A. Okay, let's see. The solution is B. It has equal amount of H plus and OH minus ions. Now, why the solution is not A? Because this question is asked like pure water is neutral since it has pH 7, it has equal amounts of H plus and OH minus ions. Water does not contain free H plus and OH minus ion, and water cannot ionize. Here, the important thing is that pure water is neutral, which is why it shows pH 7. But the reason why pure water is neutral is that because it has equal amount of H plus and OH minus ions, right? So the cause is this one, and this one is the result. So here it is asking the cause, so which is why you should answer pure water has equal amount of H plus and OH minus ions. Is that clear? Okay, moving on to the next question. Okay, you are saying that the solution is B, right? It is right because pKa is minus log of Ka, right? Ka is the dissociation constant. So when the pKa increases, it means that the dissociation constant decreases, and if it decreases, it means that the dissociation decreases, which means the acid is weaker acid. Right? Correct. Okay. Next question. What is the concentration of H plus ion in pure water? Okay, so the correct answer is C, because pH of pure water is 7, right? So minus log of hydrogen ion concentration is 7, which means that hydrogen ion concentration or H plus ion concentration is 10 to the power minus 7 molar, which is C. Correct answer. Next question. This involves a little bit of calculation. Please try to do it. Orkopal has given answer, Nandakumar has given answer. Okay. Any other answer? Alright. So let's see the correct answer. It is A. And I will just give you a brief idea of how to solve this particular problem. So are you familiar with this term, ionic product of water, which is nothing but the concentration of H plus ions multiplied with the concentration of OH minus ions, which is 10 to the power minus 14. Alright. So here the hydrogen ion concentration is given, H plus ion concentration is given. So how do you calculate the OH minus ion concentration? You will be getting it by dividing 10 to the power minus 14 with 1.3 into 10 to the power minus 4 molar. Right? So if you solve it, you will get approximately 0.769 into 10 to the power minus 10 molar, or you can write it 7.7 into 10 to the power minus 11 molar. So that is your answer. So this knowledge comes from the ionic product of water, which is the concentration, the multiplication of the concentration of H plus and OH minus ions, which is equal to 10 to the power minus 14. Alright, moving on to the next question. Buffers are a mixture of which of the following? Exactly. The correct answer is weak acid and its conjugate base. Next question. Yes, correct. The answer is B. Your pH meter is more precise. Alright, moving on to the next question. What are the parts of a digital pH meter? Yes, so some of you are saying C, which is a glass electrode and a temperature sensor. So this is also one difference between a pH meter and a pH paper. That is, pH meter can also detect the temperature in which the pH measurement is going on, or the solution, the pH, the temperature of the solution. Because temperature the pH will also depend. Okay. So this is the correct answer, C. Now we are moving on to the next question. So there can be multiple answers in this particular question. There can be multiple answers. Answer accordingly. Yes, so most of you are saying C and D, which is the correct answer. So to prevent clogging, we should clean the electrode, and to prevent generation of this asymmetric potential, we should always calibrate the pH electrode. So the correct answer is C and D. Okay, next question. Okay. Yes, the correct answer is that the higher concentration of acid or base are required to change the pH of the buffer with high buffer capacity. The correct solution is D. Next, question number 10. So I was talking about some of the characteristics of the buffers. So these are some of the characteristics which should or should not be there. So there are again multiple answers here. Right. So you are saying that the buffers should be chemically stable, they should be water soluble, and should not interact with the biological molecules. Correct. Next question is about titration method of buffer preparation. Okay. So the correct answer here is that titration method is particularly useful when preparing any unknown buffer solution, because you don't know how much of the buffer or the acid or base you need to add. So which is why it is very useful to use titration method in this case. Okay, next question. Yes, the correct answer here is that acid A dissociates to a greater extent in water than acid B. Why? Because acid A has a pKa value of 4. So A has a pKa value of 4 and B has a pKa value of 6. So B has a pKa value which is greater than A, which means Ka of B is less than Ka of A, which means that the acid A has a higher dissociation constant, which means that acid A will dissociate greater than acid B. All right. Moving on to the next question. Okay, so here the correct answer is the ratio of the ratio of the concentration of a conjugate base over acid describes the proportions of forms of a weak acid necessary to satisfy the Henderson Hasselbalch equation. Right, so it is conjugate base and acid, solution B, which is conjugate base by acid, right? Which is the form of the Henderson Hasselbalch equation. Okay. Next question. Any other answer for this question? Okay. So here the correct answer is B. So Camila, you are saying lower temperature does not promote pH changes, but that is not true, because when temperature changes, the dissociation of the acid changes. So when temperature increases, dissociation increases. So if dissociation increases, that means then Ka also increases, which means that pKa decreases, right? So if temperature increases, then dissociation also increases, which means that the dissociation constant also increases, which means that pKa value will decrease, and which will affect in the change of pH, right? So therefore dissociation of the acid depends upon temperature, which is why we have a temperature meter, a thermometer also in the pH meter, so that it can regulate the pH of the solution, the temperature of the solution with respect to the pH of the solution, right? Okay, next question. What is the difference between tris buffer and phosphate buffer? Any other answer? Okay, let's solve it. So tris buffer, as I had mentioned, tris buffer is a Zwiiterionic buffer, right? Whereas phosphate buffer is the tris and phosphate buffer, they have different pK values, right? And the problem with phosphate buffer is that the phosphate buffer may inhibit any enzyme's activity. So the correct answer is all of these. Your answers are correct. Okay. Moving on to the next question. Please read the question carefully. It's a little long question. Please read it. I've got one answer. Okay, I've got one answer. Others also please try to answer. This question is about accuracy and precision. The first topic that we discussed in today's session. Alright, let's discuss the solution. So, a new 5 rupee coin, it says it weighs 6 gram. So, 6 gram is the what? 6 gram is the reference value or the standard value, the known value. Okay, now when this coin is being measured, the weight of this coin is being measured 5 times, and these are the 5 observations that are recorded. So, in the first weighing machine, so these are 2 different weighing machines are being used. In the first weighing machine, it is showing 5.97, 5.99, 5.98, 5.96 and 6 gram. And in the second weighing machine, it is showing 5.98, 6.01, 6.02, 5.97 and 5.99 gram. So now you have to state which of the following observations are precise and which one is accurate, which one is more precise and which one is more accurate. So now, for doing that, the first thing that you have to do is that you have to make an average or mean of these observations. So first of all, average of weighing machine A. First of all, do the average or mean, calculate the mean of the 5 measurements obtained in weighing machine A. So can you tell me what will be the average of this weighing machine A? What will be the measurements of, what will be the average of the measurements of weighing machine A? Okay, very good. The average is 5.98 gram. Okay. Now, can you calculate also the average of weighing machine B? The average of the measurements. Yes, good. The average of the measurements done in weighing machine B is coming to 5.994. Now we know the reference value is 6, right? 6 grams. So now you tell me which one is more close to the reference value, or which one is closer to the reference value. Of course, B, right? B is closer to the reference value. So you can say that the measurements taken in weighing machine B is more accurate because the measurements are closer to the reference value. So of course B is more accurate, so which gives it the option B. But we will also have to comment about the precision of the experiment. And while talking about precision, what is the parameter, what do you think should be calculated, should be measured while talking about precision? While talking about accuracy, we have measured the mean or the average. Now what will be the parameter which we should consider while talking about precision? That means, yes, standard deviation. So how close are the values to each other? So, right, so that is why you have to consider standard deviation. And standard deviation is like how much that particular measurement is deviating from the average, right? So if you calculate the standard deviation of these two, you will find that the standard deviation of A is 0.016 while the standard deviation of B is 0.021, right? So which one is lesser? Of course, standard deviation of A, which is 0.016, right? So sorry, so standard deviation of A is lesser than standard deviation of B. Therefore we can say that the measurements in A are closer to each other, so they do not deviate much from the mean position, right, or the average position, which is why the measurements of A are more precise than B, while the measurements of B is more accurate, right? So that is why the correct solution is B. And do you know, does any of you know how to calculate standard deviation? Like there are several softwares which can calculate standard deviation for you, but do you know what is the method by which you can manually calculate standard deviation? Do I need to tell you that, or do you already know? So basically for calculating standard deviation, what you do is first you calculate the mean, which we have calculated here, right? So then each point, how much it is distant from the mean, so that distance you calculate. So what you do is that the distance, and then you calculate the square of the distance, so which is the mean minus the measurement. So that whole square of that you calculate, right? So suppose your mean is coming 5.994 and your measurement is 5.97. So what is the difference? That difference, the distance square you calculate, right? Now you add up the square of the distances for all points. So there are 5 points here, so you calculate the distance square for all the points, then you make a sum of all the distance squares, and then you divide it by the total number of points. That is, total number of points here is 5. So that is how you can find the standard deviation on ST. Alright, any doubt in this question, or any doubt so far? Okay. Alright. Well, now moving on to the next question. So here the solution is B. Next question. This is a question similar, a very similar problem I had discussed regarding acetate buffer, and I have also mentioned the pKa of the acetate buffer. I'm writing once again. pKa of acetate buffer is 4.8. So this information will be required to solve this particular problem. Okay, I've got one answer. Others also please try. Okay. Those of you who have answered, can anyone tell me how you have solved this particular problem? Can you unmute and tell me? Orcopol, can you tell me how you have solved the problem? Well, if you have solved the problem, you must know. Please share it with others. Okay. Okay, let's see how we can solve the problem using the Henderson Hasselbalch equation. Okay, so pH equals to pKa plus log of base by acid, right? So here the pH is 5, right? So pH minus pKa equals to log of the concentration of base by concentration of acid, right? So pKa is 4.8, so we will put that value. So 5 minus 4.8 equal to log of base by acid, right? So therefore base by acid, you can write it as 10 to the power 5 minus 4.8, which is 10 to the power 0.2. Right? Now the total volume of the solution is given to you, which is 500 ml. So therefore if you take the base as X, then the volume of the acid will be 500 minus X, right? So now you can put it here in this particular equation and you get x by 500 minus x equals to 10 to the power 0.2, which is equal to 1.58 approximately, right? So now if you solve this particular equation, you get x plus 1.58x equal to 790, right? So 2.58x equals to 790. So x will be approximately 306 ml, right? So the base will be 306 ml and the acid will be 500 minus 306, which is 194 ml. So this is the closest answer to that. So the correct answer will be D. Alright, no doubts regarding this sum? You will be getting this kind of sums in your assignments as well. Alright, moving on to the next question. So this question has multiple answers. Please answer accordingly. Okay, right, the correct answer is A, B and C. Hippies, tris, MES, all of these are goods buffer. And the last question for today's session. It is also again a multiple answer question. Okay, right. The answer is B and C. Phosphate buffer is widely used because one, it is very similar to the physiological environment, and also its pK is in a biologically relevant range, which is very useful for using it for any biochemical reaction. Alright, so that's it for today. Thank you so much for attending. Does anyone have any other question regarding today's session or regarding the week 1 content of this course? Please let me know. You can mail me in this ID, or also you can write in the discussion forum. Please let me know if you have any questions, or else we will end today's session. Okay, if you have no questions, then let's end today's session. Thank you so much for attending. Please join again in the next Tuesday 6 to 8 p.m. With the week 2 content we will be discussing, and please let me know if you have any questions. Thank you. Good night.